Hello everyone, a pleasant day to you. Today I'm going to talk about pollination and double fertilization in plants. So what is pollination and how does it differ from fertilization? Well, pollination is the transfer of pollen grains from the anther to the stigma. So last time I've been talking about the parts of a flower. For example, I've been talking about the parts of the flower last time, and I pointed out all the parts. But what matters today now are only the stamen, which consists of the anther and the filament, and the pistil, which consists of the stigma, the style, and the ovary, inside of which you find the ovule, and inside you find the egg cell and the polar nuclei. Okay, so let's start with pollination. So pollination, I just said, is the transfer of pollen grains from the anther, to the stigma. Okay, so pollen grains are caught there by the stigma. This is the stigma, by the way. So in here, let me just first present the reproductive structures involved. So first, that's the anther, and that's the filament, together constituting the stamen. And here you have the stigma, the tube, which we call the style, and the expanded base we call the ovary, which in turn contains the ovule. In this case, the ovule that I drew is already the mature one, ready for fertilization. And it's more technically called the embryo sac, inside of which you find the egg cell right beside the micropyle. The micropyle is the entry point of the sperm cell, which we will discuss later. And the biggest cell here, right beside the micropyle, is the egg cell. And then here is a very large cell we call the central cell, but it has two haploid nuclei. It has two haploid nuclei, which together we call them polar nuclei. Now these other cells, this, and this, will disintegrate. They have no function. They will disintegrate because they just have too little cytoplasm to support themselves, though they have the same genetic makeup as the polar nuclei and the egg cell. So this will be the parts involved in fertilization, which I will be discussing shortly. Now, let me just go back with pollination. Pollination is just the transfer of pollen grains from the anther, where they are produced, to the stigma, and they will stick to the stigma. Now, there are two types of pollination. We have self-pollination and cross-pollination. Self-pollination is the transfer of pollen grains from the stamen to the stigma of the same flower. So, if pollen grains are transferred from the anther to the stigma of the same flower, then that is self-pollination. It could also be that pollen grains are transferred from the anther of one flower to the stigma of another flower, but of the same plant still. It's still the same plant. So it could be from one flower to the same flower, or from one flower to another flower of the same plant. So in this case, self-pollination preserves or maintains the same genetic makeup as in the plant, where you find the stamens and the pistil. It will maintain the same genetic composition. So if the plant bears red flowers, the resulting offspring will also bear red flowers. If that plant produces sweet fruits, then the resulting plant from this fertilization will also produce sweet fruits. The other type of pollination is called cross-pollination. That is the transfer of pollen grains from the anther of one flower to the stigma of another flower of a different plant. Okay, I repeat, that is the transfer of pollen grains from one flower to the stigma of another flower, and that should be another plant, In that case, the genetic makeup in the pollen grains is different from the genetic makeup in the egg cell, so that the resulting offspring will have a genetic makeup different from those in the sperm cell and in the egg cell. So cross-pollination is the one that produces variations. And variations are very important in adaptation. Variations are very important in natural selection. They are the raw materials for evolution. That's the importance of cross-pollination. Okay, now let's follow the process of fertilization in plants. So once pollen grains are carried to the stigma, either by any agent, it could be by the wind, it could be by pollinators like insects, bats, and birds. And if we speak of insects, they could be butterflies, moths, flies, bees, or even beetles. Okay, so once pollen grains are transferred to the stigma, one pollen grain will germinate. So even if there are several or hundreds of pollen grains that land on the stigma, only one pollen grain is needed. When one pollen grain germinates, it will grow a pollen tube. So I've drawn it here, a pollen tube. So a pollen tube will grow, it lengthens and grows down the style, and then it grows down to the ovary. Since the pollen grain originally contains 2 sperm cells, then there are 2 sperm cells carried by the pollen tube. So the purpose of this growing pollen tube is to deliver the 2 sperm cells towards the ovule. Okay. One sperm cell will fertilize the egg cell, and that will produce a diploid zygote. Note, the sperm cell is haploid. It has only one set of chromosomes. The egg cell is also haploid. It has one set of chromosomes. So when the sperm and the egg nuclei fuse, they will form a diploid zygote. Haploid plus haploid equals diploid zygote. All right. Now the zygote that will develop here will be the one that will become the embryo. And the embryo in turn will form the new plant. The second sperm nucleus will fertilize the polar nuclei inside the central cell. Since this one is haploid and this other polar nucleus is haploid, together they're described to be diploid. The central cell as a whole, therefore, is described to be diploid. So, we said a while ago that the sperm cell is haploid plus 2 polar cells which together are diploid. Then they will produce a triploid structure which we call the endosperm. All right? 1 haploid plus 1 diploid equals triploid, a triploid structure which we call the endosperm. The endosperm will serve as the food for the developing embryo. This is what you call double fertilization. Why do we call it double fertilization? Because one sperm cell fertilizes the egg cell to form the zygote, which in turn will form the embryo and finally the new plant. The second sperm cell will fertilize the polar nuclei to form a triploid structure, which will serve as the food for the developing embryo. Okay, so just to show you in diagram. So if you have one sperm cell fertilizing one egg cell, this one is haploid. There's a symbol for haploid, N. And this egg cell is also haploid, that's N. Then in the process of fertilization, meaning the union of the nuclei, this will produce a zygote, which is now diploid. The symbol for diploid is 2N. And with a series of mitotic cell divisions and development, this will produce the embryo. So if it came from a diploid structure, the resulting embryo, all the cells in the resulting embryo will also be diploid or 2N. And then with further development and mitosis, this will form a new plant, which in botany we call this the Sporophyte generation, which must be diploid, unless it also produces again the egg cells and the sperm cells, which are haploid. Right. And then the second sperm cell, which is also haploid, will fertilize the polar nuclei. Note the polar nuclei are two of them, each one being haploid. So together we describe them to be 2N or diploid. In the process of fertilization then, or union of this will form a 3N or a triploid structure we call the endosperm, which will serve as the food for the embryo. Note, there is no second fertilization in animals. There's no second fertilization in animals. There's no endosperm in animals. Why? They don't need an extra food from the endosperm. Why again? Because the food of the embryo is being provided for by the placenta in mammals. And it's also provided for by the yolk in fishes, amphibians, reptiles, and birds. So there's no need for a second fertilization in animals. This is unique in plants. Okay, so this is pollination and double fertilization. Now, after fertilization, what will happen to the other parts of the plant? You see, you also have the petals before and the sepals. Now, after fertilization, the petals will wilt and fall off. They're not needed anymore. The sepals will also fall off. The stigma and the style have done their part, so they will also fall off. Now, the ovule will become the seed. The ovule will become the seed with the zygote or the embryo inside, which came from the fertilized egg cell. And then the ovary as a whole, this ovary will enlarge very, very much. It will enlarge so much to become the fruit. So all in all now, the ovary will enlarge to become the fruit, and inside it, which was originally the ovule, will become the seed. And the fertilized egg cell will become the zygote or the embryo or the new plant. The integument, if it is planted and it grows, and then the integument here surrounding the embryo sac will become the ZYGOTE. So that's how pollination and fertilization occurs, and how or what happens to the other parts of the flower after fertilization. I hope I have shared a little today. Thank you very much for watching. Subscribe for more videos. Good day. Thank you, everyone.